Good morning. Let's start with this example 3 in the lecture notes. So last class we learned how to basically convert from DNA to messenger RNA, and then from there we convert it into proteins. And this was the example that I asked you to try on your own. Uh, let's take it up. So in this example, we are given the coding strand, and it's easiest when you are given the coding strand, right? To convert the coding into the messenger RNA, all you have to do is copy everything. But of course, you have to replace the T with U because there are no thymines on the DNA, only on the RNA rather. There are only uracil. So this would be AUG dash ACA UUA CUA GCA GGC and ACU. And then once you have the messenger RNA, you can convert it into the amino acids, right, the protein. And you would need to have the codon chart for that. Right? So again, please make sure you print your own for the quiz. And so we can look up AUG, that's the start codon, right, which is MET, methionine. Now, the start codon is at the beginning of the protein. It doesn't mean you cannot have another methionine in the middle of the sequence. Right? That's possible. It's like English, right? Writing a sentence. The first letter in the sentence has to be capitalized, but you can also have a capital letter somewhere inside the sentence, right? Like if you write a name or a place or something like that. So same thing. Okay? Just because we have to start with methionine, it doesn't mean you cannot have it again later on in the sequence. So you might have another AUG in the middle of the protein sequence, and that's fine. Next, we have the ACA. Right? So ACA is threonine. And then you just kind of continue the process, right? Hopefully you have done this already. UA gives you LEU. CUA gives you another LEU. GCA gives you ALA, and GGC gives you glycine. And finally, we have the ACU, which gives you threonine. It's also important to note that when I give you a sequence like this to transcribe and translate, it's not actual like an actual protein sequence, right? Like in real life, the protein sequence are much longer. Sometimes, hundreds and hundreds of amino acids. So maybe this is going to continue, right? So that's why this is not yet the stop codon. Okay, so it's not just because you didn't get a stop codon at the end, it doesn't mean you did it wrong. That's what I'm saying. Okay, so that's basically the key thing that you had to learn from last class. Let's take a look at this poll question. If a codon contains 4 nucleotides instead of 3, what is the maximum number of amino acid such codon combinations can code for? So
256.
exactly, very good, thank you. So you just put 4 blanks, and it's like 4 choices for each blank, right? A, C, U, or G. And then you multiply them together, you get 256. Now that partially explains why the codon that we use actually consists of 3 nucleotides. It cannot be 1 nucleotide. If it's 1 nucleotide, we only have 4 choices, A, C, U, or G. 4 is not enough to code for 20 different amino acids, right? So what about 2 nucleotide? Can 2 nucleotide makes up enough codons? Well, if we do 4 times 4, that is going to be 16 codons. But again, we have 20 amino acids, right? Having 16 codons are not enough. And that's why we have a triplet. Right? Triplet gives us 64, and it's more than we need, and we don't need to go even higher. Okay, and it's good. You will see today why it is a good idea to have more codons than we need for amino acid. It's actually a protective mechanism against mutations. Okay, let's take a closer look at the molecular level. What exactly is going on with the process of translation? How exactly, do you connect the amino acids together? Now, a long time ago, I already told you we have 3 different types of RNA, right? There is the messenger RNA, and you know what that is by now. That is the transcript, right? A copy of the gene that contains the instruction to make the protein. You can think of this as like the cooking recipe. Cooking recipe. It tells you how to make the dish. Then we have another one called rRNA, ribosomal RNA. This is the type of RNA that makes up the ribosomes. Remember the ribosome, the 2 subunit, the big and the small, and the ribosome is made of both RNA as well as protein. Okay, so the kind of RNA that makes up the ribosome is the rRNA, and this kind of RNA is important for linking the correct amino acid in the proper sequence. So this is the chef that's going to be cooking the recipe. It's gonna be looking at the recipe, looking at the mRNA, and then find out what to do, what to put together to cook, right, to make the protein. Then this is the new kind that I haven't really told you about. Right? This is called tRNA, which stands for transfer RNA. Transfer RNA. Transfer RNA is important for bringing the correct amino acid to the ribosome. So you can think of this as the assistant, okay, the helper of the chef. Like the chef is like, hey, I need this, I need that, bring it over. That's what the tRNA does. It brings over the correct amino acid, and then the rRNA will join them together. Do you understand? Right? The instruction is in the mRNA. The tRNA brings the amino acid over, and then the rRNA connects the amino acid together. So together, these 3 different types of RNA plays an important role in the process of translation. Okay. Let's take a closer look at the tRNA. The tRNA looks like this. It's like a clover leaf, right? Like a club, if you would, and it has 2 ends, okay, that you have to pay attention to. At one end here, this is where you will attach to an amino acid. So amino acid attachment site. Okay. So you will hook an amino acid onto it. And then, of course, since this is RNA, you would have a bunch of nucleotides, right? Like, A, C, U, G, whatever, right? Okay. And what's interesting is the RNA is single-stranded, but in this case, it's able to fold back on itself and form bonds with another part of the RNA. So for example, I'm just making this up. Let's say this is ACUUA or something. Then at this part, it is going to be AAU, right? Because the sequence are complementary, it's able to fold back on itself and then form a stable structure. And so that happens in 4 places. It happens like up here, on the sides, and at the bottom. Okay. The most important thing that you have to understand is at the bottom here. At the bottom, there is a triplet of co that is particularly important. I mean, the whole thing is full of A, C, U, and G. But right at the bottom of this cloverleaf structure, there is a triplet that is particularly important. And we call that the anticodon. The anticodon. The anticodon is complementary. Complementary to the codon on mRNA. And the anticodon determines which amino acid the tRNA carries. I'll explain more in a bit, but just, copy down what I have here first, and then I'll give you an example. Oops, sorry. So let me go over here, and we actually gonna have a bit of nooks today. Maybe, I don't know. I forgot what's on the PowerPoint slide. We'll see. All right, you want to copy this stuff. So let's say I have a messenger RNA. Okay, messenger RNA. And then 5' to 3'. Now, and I will have some codes on it. Okay, I will have some code on it. AUG, UUU, UGC, CCA, GUG, and then UGA. Okay. Just copy that first, and I'll tell you what this is about. Now, as some of you are copying, some of you may be done, can you tell me what amino acid AUG corresponds to? Just look it up. Or like you probably remember it by now if you have been practicing. What does AUG code for? Yeah, that's right, MET, right? So we're gonna have our ribosome here. See if I can have more colors. Where's brown? Brown. Okay, so we're gonna have the ribosome here. Okay, and then the ribosome is made up of rRNA, right? And protein. And then we're gonna have our tRNA, our transfer RNA. So there are several transfer RNA here. I'm just gonna draw a few here. Okay. So let's say this one is carrying MET, and let's say this one is carrying PHE, and then you might have a few other ones. Doesn't really matter. Let's draw another one here. That's like ALA or something. So all these are amino acid, right? Now, which one is going to be the correct one to bring the amino acid for the first codon here? Well, we looked up AUG on the codon chart, right? Okay, so this one is gonna come over here. It's gonna come over here, like that. Okay. But how does the tRNA know? I mean, like they don't have a codon chart to look up, right? Like we, when we did research with scientists, when humans do research on how this works, they find out about the codon chart, right? Okay, but like, how would the tRNA know? Right? Like this thing here, you know, let's say we choose the wrong one here. I know it's difficult to draw, okay, but just try to follow what I'm saying. Let's say this over here, like is it gonna not fit or something? Like what is the mechanism? What is the method to tell one tRNA apart from the other, right? Well, it turns out the answer, I already told you, has to do with the anticodon. So over here, I actually have to look it up so I don't make a mistake. I have to look at the codon chart. Give me a second. So this is so this would be like CGG, for example. Is that right? Give me a second. GCC. Yep, that looks right. Okay, and then over here, I would have a UAC, and here I would have like AAA. Okay, so let's remind ourselves that's the anticodon at the bottom. Okay, like I said, the whole thing is made up of ACUG, but then there's a special triplet at the bottom. That's called the anticodon. Yeah, like magnets, you will see. Okay, so like let's say this one PHE, right? It comes over here. Just flow and try to fit here. Like it's not gonna fit. Why is it not gonna fit? Well, because AAA is not complementary to AUG, right? So it's not gonna fit, and then it's just gonna bounce away. Okay. And then what about this one? Let's try to go over here. Okay, and then it's complementary this time, right? They pair up. So if everything matches up, then you will deposit the MET. Right? So like this MET is gonna stay here. Okay. I know it's very hard for you to draw because I'm like moving things around. Maybe you don't need to draw it. So this thing is gonna go away and then just leave the MET here. Okay. And now this is empty. This is empty. This is missing and methionine now. So there are a bunch of amino acids that are just like floating around in the cytoplasm. And what it's going to do is basically just going to reload itself. So put another one on, and then we can deliver the MET to another location. Do you guys understand what I'm saying so far?
So like the clover things are just like cars bringing things to places, and then like the codon charts are like the people getting in and out.
If that works for you, sure. But the
Okay.
tRNA is more like, again, like the little helper, right? If you use the cooking analogy, it's bringing the ingredient so that the ribosome can put them together, right?
Okay.
Okay, so guys, help me out here. The next one is UUU, right? Okay. Without even looking at the codon chart, which of these should come along? Here, I'll draw another one here for you. This one is like CCC. Okay, so which one is gonna come? Yeah, so like UUU must match the anticodon AAA. So it has to be this guy. So this is gonna bring over here, you know, like that. And then it's gonna go. It's gonna go away. And then now I would have another empty one. Empty one here. Does that make sense? You guys got to give me something like
Is it the same clover or is it a different
oh,
clover
it
that
could be
goes?
like yeah. So like you would have a bunch of these clover leaves, right? A bunch of these tRNA that would have the AAA. Like it's not
Okay.
just one, right? It's not just one helper, if you would, right? Like I would have another one here that's also AAA, right?
Okay, so you can just go like die off whenever and no one cares.
Yeah, but they're not gonna die off. They they would go pick up another MET, right?
Mhm.
Actually, I got the wrong amino acid here. This one should be PHG. Right? And then it would bring it over, and then afterwards it would go get another one, and then it doesn't have to even come back to the same ribosome. You have so many things building proteins at the same time, right? Do you
But once
understand?
it has like CCC, it can't change and have like a UAC or.
Exactly, right? So the clover leaf themselves don't change. The anticodon is imprinted with them. So this particular tRNA can only carry PHG, right? So that's what I mean. In the PowerPoint notes, I think it's probably gone by now. The anticodon determines what amino acid it's carrying, and it can only carry that one and nothing else. Does that answer? Is that your question, or am I totally off here?
Yeah, I'm just double checking.
Oh, okay. Yeah, like Legos, right? Like I need to build stuff. Okay, so bring me a red piece, bring me a yellow piece, bring me a green piece. Okay, whatever analogy you need to help you understand this. Okay, let's just really hammer the point and do it one more time. So the next one, right? The next one, the codon. You gotta understand the difference between the two things I'm talking about here, right? There is the codon and then there's the anticodon. So if you look at here, sorry, I just gotta choose my blue color here. If you look at the codon, oops, if you look at the next codon, right? What kind of tRNA do we need? What kind of tRNA do we need? What should be the anticodon at the bottom here? I want the anticodon first. ACG is correct. Okay. And then what kind of amino acid will it carry? Now remember, the chart, the table, let me just pull it over. Like this table, this is for codon. You never ever look up anticodon. If you're looking up anticodon on the test, you are doing it wrong. Okay, you always look up the codon. Please remember that. So you're gonna look up UGC. Right, so UGC is cysteine. Right. So again, this guy's gonna come over, pop into the slot, and then it can go away. Make sense? Yeah, I'm gonna get their medicine. Hold on. So this is CYS. Now, I'm sorry, I'm like erasing a lot. You must be feeling frustrated if you're copying this. But like I said, maybe you don't have to copy, right? Where's my brown color? Was it this guy? Okay. So what happens is after you finish building the first three, you're gonna shift over. You're gonna shift over a little bit. Right? It's gonna move along, and then you just keep on doing it, right? People sometimes call this like imagine like eating a spaghetti. Okay, so like the ribosome, and then this is the spaghetti. It just keeps on moving forward, and then it reads the code as it goes on and on and on. When it gets to the stop codon, there will be no tRNA corresponding to the stop codon. And everything just stops. That's the signal that tells the ribosome, it's done. Thank you. We are finished. And the protein is gonna pop off and it's finished. Okay? Now let's take a look at this with the PowerPoint slides. Maybe it's gonna be more clear. Number one, amino acids and tRNA float freely in the cytoplasm. The tRNAs are always there. Amino acids you get from breaking down your food, right? You eat your proteins, you catabolize, if you remember that word, right? You break down the protein, and then you get the amino acids. They're just floating around in the cytoplasm. And then an enzyme will help put the amino acid onto the tRNA. So over here, there is an anticodon of GAG. Which amino acid should this carry? What amino acid should this tRNA carry? Anybody? What are we going to look up on the codon chart? Are we going to look up GAG, guys? Okay. Ramon said ARG. Well, you have to remember this is the anticodon, right? Anticodon. So when you try to figure out which amino acid it's carrying, you have to think about the codon. So this is the mRNA, the codon. Yeah, thank you, Roy. It's CUC that we are looking up, right? Remember, I told you literally, I told you 2 minutes ago, do not look up the anticodon. If you're looking up the anticodon, you are doing something wrong. So if we look up CUC, you will see that it is LEU. Okay, so this is going to be right here, LEU. Are we okay? Let me know, guys. If you don't understand, I can explain again. Because like otherwise you get very confused for the test, right? I don't want you to do it wrong on the test. You get it? You don't get it? Yeah. Okay, thank you. Can you explain again? Sure, sure. Okay. So, is it Sarah? Okay, Sarah, I'm gonna ask you a question, okay? What do we call this triplet again? What is the name of this triplet at the bottom? Yeah, the anticodon, right? Okay. The anticodon is going to help decide what amino acid we are going to be putting on to the tRNA. Okay. And ultimately, the tRNA will be bringing the amino acid to the messenger RNA, right? So what kind of code will AGU be binding to? If you think about the messenger RNA, what is the codon that will be on the messenger RNA in this case? Like think about this blue ribbon, right? Okay. What would be the codon on it so that it's going to fit? That's right. And you got that because of base pairing rule, right? So that's UCA. And that is the codon. So we're going to look up the codon. We're going to look up UCA on the table. And what does UCA code for? That's right, SCR. Is that better? Anyone else have other questions? All right, so step one, we gotta load the tRNA. If the tRNA is empty, if it doesn't carry any amino acid, is useless. It's not gonna help us. So we're gonna load them up, and based on the anticodon, we can figure out the codon, and then we can figure out what it's supposed to be carrying. Then this is what I was trying to draw for you. Right? Basically, what happens is this green chain here is a. Can you guys see my mouse, by the way? Like I've been having trouble lately. People cannot see my cursor. Can you see my cursor moving around?
Yes.
Okay, great, thank you. So like this green chain is the amino acid, right? That we are building, right? And you can tell, we have been building for some time now, right? Like probably at the very beginning here, there's like a MET somewhere, okay? Like we don't know, but like, long time ago. But basically, what needs to be happening is that the tRNA will dock if complementary RNA codon is present on the ribosome. In other words, we cannot just randomly put the tRNA into the ribosome here. For it to be the right tRNA, it must be complementary, right? To write it better, the anticodon on the tRNA must be complementary to the codon on mRNA. Okay, perhaps this is like the key sentence that summarizes, the most important thing. Once you break the amino acid join together to form a polypeptide, right? And what's allowing them to join together? It's the job of the ribosome, okay? The chef, right? It's like linking the stuff together. And then the empty one is gonna exit. Where is this gonna go? Well, this little guy right here, this is going to go and reload. Right? It's gonna pick up another amino acid. Okay. And then it's gonna come again. And you can see the ribosome continues to move forward. Right? So new one comes in, deposit the amino acid, and then it leaves. And then another one come, and then it leaves. It's just like, clockwork. Keep going, keep going. Until when you get to the stop codon, okay, when the ribosome reaches the stop codon, no tRNA can base pair with the codon, and then the whole thing stops. That's the cue for the ribosome to finish its job and release the RNA, release the protein, and then we are done. That's all. Okay. Let's try this question. We launch the poll. I only have 3 respondents. Can we have more, please? Okay, 6 now. Anyone else before I give you the answer? Oh, that's okay. Yeah, don't do the polls if you're driving. Stay safe. Yeah. Okay. Well, this is the anticodon. This is the anticodon. So, what do we look up? We don't look up AAA, right? we look up the complementary codon. We look up UUU. UUU is phenylalanine, PHG. Okay? You will see some questions like this. So make sure you just, just remember my voice telling you, if you're looking up the anticodon, you're doing it wrong. All right. So this slide tells you how we can control gene expression. I already told you before, even though all the cells in the body have the same set of genetic material, same set of genes, you don't turn on everything in every cell, right? Depending on the function of the cell, you need to turn on a specific group of enzymes. Sorry, genes, right? Particular group of genes. So how do you control it? Well, you can actually control it in several ways here. You can control it. The easiest way, the most straightforward way of doing it is transcriptional control. You control it at the transcription level. You don't want to turn on the gene, then don't transcribe it. Okay, don't copy the DNA, then you won't have the mRNA. It's completely off the gene. Okay. But even after you transcribe it, you can still modify it, right? So we call that post-transcriptional modification. We know in post-transcriptional modification, we can add the 5' cap and the poly A tail. We can remove the introns and keep the exons. So I can control how long the tail is, and that would control how long I will have the messenger RNA, right? Now, I didn't really tell you this before, but it turns out you can pick and choose what exons to keep, right? So if you decide to keep exon 1, 2, 3, 4, the protein might be a little bit different than when you decided to keep exon 2, 4, 6 only. Okay, so you have a little bit of control there. And even afterwards, you can control it by the process of translation. There are mechanisms that dictates how often and how rapidly, like how fast, you are gonna translate the mRNA. You have the ingredient there, but it doesn't mean you have to make it super quick, right? You can do it slowly, okay? Or you can do it quickly, depending on how much you need, right? And how often do you do it, right? Do you just keep on making it, or do we make it on demand, right? So there are mechanisms to help control the timing of it. And afterwards, even after you make the proteins, you can still control it. Okay, you might remember the Golgi body, right? The Golgi body will receive the vesicles that contains the protein that is freshly made, and then you can sort them, you can modify it, you can repackage them, and you don't necessarily need to send it out right away. Right? You can keep it in the cytoplasm until you need the protein. So there are all these different levels in which how you can regulate the expression of the gene. Right? It's not like once you turn it on, then you have to use it right away. There's so many levels, so many layers of control that are available. All right. Now we're going to move on to mutation, which is another could be confusing. It could be like a lot of work. So pay attention. What is mutation? Mutation is permanent changes in the genetic sequence of an organism. It's a mistake that was not fixed, basically. And I told you in the last class, the mutation can happen spontaneously, just bad luck. You copy, if you divide your cells enough times, if you copy your DNA enough times, there is a high chance that you're gonna make some mistakes, right? Or it could be induced exposure to carcinogen, mutagens, right? Environmental factors. Changing the genetic sequence might result in change in amino acid sequence. Okay. How do you prevent mistakes? Madison asked. Well, for the spontaneous type, you can't. Right? Like I said, it's just bad luck. It's bound to happen. It's imagine typing an entire book of encyclopedia over and over again. right? If there's no spell check on the, even if there's spell check, right? Like you are gonna make a mistake eventually, right? Because there's so many words, and you know, you might miss a word here, you might misspell a word there, and even with spell checking, you might still have a mistake, right? That's just inevitable. It just happens. But for the induced one, right, we can limit our exposure to mutagens, right? So people say, you know, don't smoke. You know, go away if there are other people smoking, so you don't inhale secondhand smoke, right? Use sunscreen, right, to protect from UV. So all these things, right, that we can minimize environmental factors, environmental triggers. But you know, even if you do everything right, mutations could still happen. It's just part of being, you know, humans, I guess. So changing the genetic sequence might result in changing amino acid sequence. I'll tell you why that is the case. It's not guaranteed. Just because you change the genetic sequence, it might have no effect. Since the protein function is dependent on the order of amino acid, mutation can alter how these proteins function. We already talked about this in the last lecture. You might have a gain of function mutation in a proto-oncogene. You might have a loss of function in a tumor suppressor gene, and that could completely change how the cell behaves, right? Now, some of these mutations could be hereditary. You could pass it on if it happens in the gametes. Okay, but if you acquire a mutation that is in your bodily cells, in your somatic cells, these are cells that are not sperm and egg, then only you will experience the effect. It cannot be passed on to future generation.
A quick question.
Yeah, sure.
So you said the protein function is dependent on the precise order of amino acids. So if the amino acid sequence changes, then the protein function will change, right?
That's right. That's right.
Okay.
But it also depends on what kind of changes. I'll explain. I'll explain more in a bit. Okay, so let's talk about mutations here. How should I do this? I'm just thinking if I should do it on a note or if I should. You know what? Let's do it on a note. Why not? It's probably cleaner that way. Um, we'll come back to this. I don't know if you guys want this thing or not. Should I erase it? You know what? I'll just keep it. Um, you know, it's just chicken scratch over there. It looks like very messy, but hopefully it conveyed the information. Now, you will need this chart with you for this next part, okay? So grab yours on the phone or whatever. If you printed it out, fantastic. So we are talking about mutation here. Mutation. So mutation comes in several forms. All right. We're gonna look at what's called base substitution first. Base substitution. Okay. This is also called like point mutation. It happens at a single point. Okay. This is when a single nucleotide changes. Okay. There are three possible outcomes. Now, I probably should write this down for you. For changes to be permanent, the mutation has to occur in the DNA. But to make it easier to understand, we will give examples as changes in RNA. Okay? So the changes has to happen in the DNA. If the changes happens in RNA, all right, listen for a second. Okay, stop writing. Listen to me. If the changes happens in RNA, then it's not permanent because the RNA will degrade after a while, right? When you do another transcript, it's gonna be fine. Right? It's only when the mutation happens in the DNA, then it's gonna be forever. It's gonna stay there, and all the cells after that is gonna be affected. Okay, but since we're just trying to understand the process, it's really hard to do it straight from a DNA at the beginning. That's why I'm giving you examples that are already transcribed. Do you understand? The examples I give you is like, we know it happens in the DNA. Let's look at what happens after you do the transcription. That's what this red little blurb means. Okay, copy it, and then when you're ready, I'll give you 3 examples. Okay, here we go. First example, neutral mutation. Let's say we have the original code is UUU. Okay, so this is the original codon. Okay, we call this the wild type. Wild type. No change. That's what, like, most people have, for example. And let's say we have a mutation. And remember, I know I've said this like a couple times already, but the mutation happens in the DNA, right? Okay. After you make the mistake and then you do the transcription, let's say the mutation caused this to become UUC. UUC. Just gonna move this guy out of the way. So please look up UUU and look up UUC and tell me what they represent. Thank you. That's UUU, right, Ramon? That is PHE. Very good. What about UUC? Give me UUC. Yeah, UUC is also PHE. All right, look here. UUU and UUC. They are both PHE. Okay. So what's gonna happen? If you have this mutation, what will happen? What's the result, huh? Nothing. That's right. Nothing happens. This kind of mutation is silent. You wouldn't even know that you have it. Okay? Because it didn't change anything. It didn't change the amino acid. You didn't change the protein. It's the best kind of mutation to have. Right? If you're going to have a mutation, you better hope, you better pray this is the mutation because it's not going to affect you in any way. Okay? Quick question. Between you and me, okay? How much alike is our DNA? Give me a percentage. How much of our DNA are the same? Give me a percentage. Or you can say how much is different. Like you and I. 50%? Yeah. We are not related. I hope we are not related, gosh. All right. So like is it just 50%? Okay. Anyone else? 1% difference? Now we're getting somewhere. Anyone else? 99, yeah, okay. You and me, or for that fact, you and any other person in this world, doesn't matter their race, doesn't matter if you're related or not, because we are both humans, we are 99.9% identical on the genetic level. Okay, it's hard to believe, right? It's very, very similar. Okay, so that 0.1, sorry, that 0.1% difference between any two individuals, a lot of it actually has to do with this neutral mutation. Okay, we actually call these SNPs, SNPs, single nucleotide polymorphism. Okay, we'll learn more about that in lecture 9. Okay, polymorphism just means differences in the genetic information between two peoples. So these are differences that exist on a single nucleotide base, right? Isn't it like cool? Like a lot of people actually don't know that, right? They think, oh, you and I look very different, right? We must only share very little genetic information. Okay. What about humans and chimpanzee? What do you think? Humans and chimpanzee. A good amount the same. It's about 98.8%, 98%. Okay. Yeah. Did you look that up on the internet? Yeah, Ramon? Yeah, that is that. Yep. Good job, right? That's the chimpanzee between us and chimpanzee, right? We are so much smarter than the chimpanzee, right? But on the genetic level, it's so different. It's not so different. It's very, very similar. Okay. Amazing stuff. Anyway, back to this mutation business. Yeah, exactly, right. 1.1%, that's so much. Like I recently read a book, and it was saying this is just random stuff. Okay, I when I read it, I thought it was pretty cool. Right, the book was saying that between us and the chimpanzee, it's only extra 1.1% difference, right? Okay, 1.1, 1.2% difference, and yet the smartest chimpanzee. What can a smartest chimpanzee do, right? The smartest chimpanzee can maybe do a little bit of sign language, okay? But like no matter how hard you try, the smartest chimpanzee will never be able to do simple math, like even addition or stuff like that, right? Okay. So imagine if there is another alien species that's like 1.1% DNA difference from us, but like on the next higher level, right? Just like the chimpanzee, the smartest chimpanzee will not be able to do any basic addition calculation that even 3-year-old humans, 4-year-old humans can do, right? That alien species, right? Maybe their babies will be able to do like calculus, right? And it will be very easy for them, right? So that just 1.1 difference, that extra step, that new next step in the evolution can really make a huge difference. Okay. We can't do the same thing as those aliens. Yeah, basically, right? Like if they are like next level up, right? In terms of like the DNA difference and in terms of intelligence, right? Anyway, I don't know. I read that book and it was pretty cool. It's called Astrophysics for busy people. If you want to look that up. Anyway, I went on a tangent. Let's go back to biology. So there's the neutral mutation, and then the next one is a missense mutation. Okay. By the way, like I said a lot of stuff, but the key point I was trying to make is neutral mutations doesn't do anything to change your protein, and it accounts for some of the differences that we have between human beings. Okay, that's the SNPs that I was talking about. So let's do missense mutation. Let's say we take like UUU, and this time we mutate it to UUA. All right, so look up UUU. What is What is UUU? We already know it's PHE. And then UUA, right? UUA is up here. Right? UUA, right? So that's LEU. Right? So that's a change. That's a mistake. That's an error. That's why we call this missense mutation. Now, is this going to be bad? Well, that depends. That depends. Right? Let me try to write it out for you. I gotta find the right color again. So that depends the result, right? Depends on where the amino acid is in the protein. Okay. We already learned that the sickle cell anemia, for example, just one change is enough to cause the disease. Okay, changes in amino acid number 6. But it really depends on, well, it might not be cancer, right? Like not everything is related to the growth inhibitors or growth promoters, right? Some are just enzymes, okay? So let's say we look at an enzyme. An enzyme is a protein, and you might remember the enzyme. Here's an enzyme. This is how I used to draw it, right? The enzyme has an active site, right? Okay. And remember, the enzyme is protein. The whole thing is made of amino acid. So what if this mutation happens like right here at the active site? What if you change an amino acid right at the active site? Well, that could change the shape of the active site. Remember, how the protein folds together depends on the sequence. Right? So if you change the amino acid, if you have a missense mutation in the active site of an enzyme, that could change the shape of the active site and cause the enzyme to no longer function. So that could be bad, right? However, what if the mutation is like somewhere else in the enzyme that's not in the active site? Maybe it's just like at the end of the protein. Maybe it's like at the site, at some non-important site. Then maybe it's okay to have this mutation, right? So it's dependent on the location, depending on where it happens. Okay, just think of I guess your car, right? Like if you're gonna have a damage, like is it gonna be bad? Well, it depends where it happens, right? Like if you run over a nail, okay, the nail punctures your tire, then your car cannot be driven, right? Okay, because you have a flat tire. But what if that nail, got inserted into your trunk? I mean, it's not going to be pretty, but hey, it's not going to affect the functionality, right? Same problem, same damage. It's the nail, but depending on where it happens, the effect, the outcome is going to be different, right? So same thing for this missense mutation. Right? Depends on where it happens. It's not always bad. It could be bad. Okay. Now, last one here. Last one. The last outcome is nonsense. Nonsense. You might be thinking, hey, all this stuff that we're learning is nonsense. It doesn't make any sense. This one is, let's say, UGC becomes UGA. Okay, please look it up. Tell me what's going on. Okay, UGC is medicine told me is a cysteine. Confirm that, guys. And then what about UGA? UGA is stop. Okay, so what's the result? The result is a premature termination. termination of protein synthesis. Okay, you have what's called a truncated. Truncated means shortened. Okay. So imagine you're supposed to make, you know, a protein that's this long. These are amino acids, by the way. Okay, and you have a nonsense mutation. So is it bad? Again, you have to know where it happens. If it happens like right here, let's say the nonsense mutation happens here, that's pretty bad because the rest of it is not gonna be made. You don't even have a protein. It's gone. Right? That's pretty bad. What if it happens in the middle? Yeah, that's pretty bad too. Like half of the protein is missing. Basically, the earlier it happens in the sequence, the worse it is, right? So when is it not too bad? Well, if it happens at the end, maybe it's okay, right? Because you're gonna lose a few amino acids. Maybe it's like the bulk of it is still there and it's gonna be fine. Does that make sense? Right. So like if you think about this as a tumor suppressor gene, right? If there is a nonsense mutation that happens at the beginning or even the middle, that's totally a loss of function mutation. Right? The red light's not working. It cannot work if the red light is not there, right? If you're not making it, it's not gonna show up. It's not gonna do its job. So that's an example of how loss of function mutation can arise. Okay. If you are a mother, when you found out you were pregnant, you probably did some blood work. Okay, and one of the things that they test for is, and often they not only do they test your blood, they also test your spouse blood in most cases. So one of the things that they screen for is something called beta thalassemia. Okay, so beta thalassemia is another type of anemia. So you know about sickle cell anemia already, right? I taught you that, where the hemoglobin misfold. But in beta thalassemia, one form of it is caused by a nonsense mutation. So the hemoglobin, which has 4 chunks to it, you're missing a whole part because of the nonsense mutation. You didn't make the protein basically, right? And that has hereditary basis to it. Right? Even if you don't have it, you could be a carrier for something like that. Okay, so they want to assess the risk of your child having this problem. Okay, so 3 types. These are point mutations. These are base substitution. You're just changing one. Now, in my examples here, right? Like I'm only changing the last letter here. Okay, but it doesn't have to be like that. You could change any of them. You could change the first one. You could change the middle one. All of those are going to be, point mutation. Now, just one last thing before I let you go on the break. This explains why having more codons than amino acid is actually a protection against mutation, right? It is precisely because we have multiple things that codes for the same amino acid that we can allow neutral mutation to occur. You see, the ones that are the same actually usually happens within the same group, right? So you see, like CCU, if you mutate to CCC, you are okay. If you mutate to CCA, you are okay. If you mutate to CCC, that's okay as well. That's like 4 mutations. That's going to do nothing for you, right? It's a protection against mutation. Right? Same thing here, right? And you can get more here, right? So all these groups that are together, it's protection against mutation. Even if you change the nucleotides, there is a chance that you are going to be okay. Any question before we go on the break? All right, I'm gonna leave the screen on this note. You copy whatever you need to. If you think of anything else to ask me, you can. When we come back, we will learn the other type of mutations, and then we're gonna spend the rest of today doing practice, okay, from the study guide. Okay, see you in a bit. Okay, let's keep going. Let's go back to PowerPoint slides for a bit here. So this is the note that we took. I'll post it online, of course. Missense, nonsense. And here is an example that shows you again the mutation must happen at the DNA level. Right? So that's the DNA, right? That's the DNA. So over here you can see if you have different, there are 3 mutations here, and the consequences of each. Right? So they did the transcription, which gives you the mRNA here, right? AUG, right? And you can see that the coding and the mRNA are the same, right? ATG becomes AUG, and that codes for methionine, and so on and so forth. So the first mutation here, instead of the wild type TNA, right, they mutated to CNG. And because CCU and CCC both codes for proline, no change, silent mutation. And then over here, right, because of the changes in the DNA level, it changes from the A and T. It changes from the T and A here to A and T. They flip it. Okay, that causes a missense mutation. So instead of arginine here, you get a different one, histidine. And if you change this here, CG to AT, then you get a stop codon, a nonsense mutation. Remember, there are 3 stop codons, right? UGA, UAG, and UAA. So anytime you change it to one of those, it's going to be a nonsense mutation. You're going to create a truncated protein, a shortened protein. We already talked about this situation, the sickle cell anemia. Now, that actually reminds me to talk about another thing. Sometimes you can have a missense mutation that doesn't have too much of an effect. I already told you, if it happens in like a non-important part of the protein, it's fine, right? But it also depends on the property of the amino acid. When we learn amino acids in lecture 2, I show you a big table with all the different amino acids in it, right? The 20 amino acids, and they can be grouped together based on their properties. Some are polar, some are non-polar, some are acidic, some are basic. If the mutation substitute a polar amino acid with another polar amino acid, maybe it's okay because they have similar property. Right? In the case of sickle cell anemia, because the mutation changes the polar to a non-polar amino acid, right? The change is so drastic that a single mutation is enough to cause the disease. All right, let's take a look at the next one here. Another kind of point mutation is insertion or deletion. So in this case, we are not changing the amino acid. We are not changing the nucleotides. Okay, in the base substitution, we replace a nucleotide with another nucleotide, right? In this case, we are not doing this. We are inserting extra nucleotides, or we are deleting nucleotides. Okay, and this could happen spontaneously when the DNA polymerase is trying to copy a section that has a lot of repeats in it. Okay, so you already know that repeats are common in DNA, right? For example, at the ends of DNA, we have repeats of TTA, GGG. But sometimes, like within the DNA, we have repeats that are very similar. Okay? For example, like repeats of like CGC. Okay? CGC. So it could repeat like 50 times. CGC, 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 like 50 times, right? So when the DNA polymerase come copy it, it's very easy for it to make a mistake. Like you would too, right? If I asked you to type a bunch, copy a page that, with a bunch of CGCs, right? And then, you know, maybe a bunch of ATT. When you type it, it's possible that you miss a few, or like maybe you insert an extra one, right? Without realizing it, right? So that causes insertion or deletion mutation. Insertion and deletion can lead to what's called frame shift mutation. If you are adding or deleting nucleotides that is not multiples of 3, that caused the reading frame to change completely. What does that even mean? I will give you an analogy and it will help you understand. Let's say we can only read sentences in triplets. Okay, so here we have a sentence. The big cat saw the fat dog. Okay. The sentence makes sense, no problem. I only read in triplets. The big cat saw the fat dog. Now, what if we deleted the B? Remember, the rule is we can only read 3 letters at a time. Right? So if we deleted the B, then the sentence becomes the, which is fine. It's the same word. But then everything after that is gibberish now, right? Like IGCATS, right? Once you deleted the B, and we can only read in triplets, the next thing you read is IGC, ATS, AWT. Do you understand? Everything here is messed up, right? Because we shift the reading frame. We have a frame shift mutation. Now, that's not going to be a problem if I delete the whole word big. Okay. What happens if you delete big? No, the sentence still makes sense. The cat saw the fat dog. Okay, we lose a bit of information, but for the most part, the whole thing is still like it still maintains, right? The sentence structure, right? What if we add an extra letter? Same problem. Once you add an extra letter, you shift the frame, and nothing makes sense. What if I insert 3 letters? Right? Now we have extra information that wasn't there before, but again, because we inserted 3 letters, it didn't mess up the reading frame. Since the ribosome, I should say, since the ribosome reads the messenger RNA in codons, and codons exist in triplets, that's why anytime you insert nucleotides that's not a multiple of 3, you will shift the frame. If you insert 3, it's okay. You have extra amino acid. If you insert 6, that's okay. You have 2 extra amino acids. I mean, like it's okay in the sense that you will still cause a mutation, but it's not as bad as a frame shift mutation. Okay. So this is an example. I think it's better if we work on an example together. Okay, so please take out your study guide. Study guide is right here. And I want to go to this exercise right here. Okay. Now, if you don't have this, that's fine. You just copy it, okay? Now, it's better to chop it up, like this into triplets. So this one here is the the template strand, it says, the template strand. So what we're gonna do is we will make the amino acid sequence. Since it's template, we have to use the base pairing rule. So that would be AUC. Okay, put a line to separate it. And then, you know, the usual. Now to save you time, I will give the amino acid sequence to you. I strongly recommend you do this on your own when you have time. Okay, this is nothing new. This you should know already. We learned this last class, right? Next, we have a mutated DNA sequence. So again, you can chop it up into triplets. And we want to identify the mutation. Where is the mutation? Do you guys see the mutation? Where is the change? You have to compare nucleotide to nucleotide. TAC, TAC, so no problem. ACC, ATC. There's my mutation, right? There's the mutation. So with this new mutation, go ahead and do the mRNA sequence and then find the amino acid sequence. I'll give you a moment to do that. Okay, please try that now. So everything is the same. You can just basically copy it, right? From above, AUG, and then instead of UGG, it is now UAG, right? Because of the mutation. And then the rest, I mean, you can just copy again. Right? So what happens? Well, let's do the amino acid sequence. AUG is MET, and then UAG, stop. Uh-oh, we are not making the rest of the protein, right? Will there likely be an effect? Yes. The whole protein is gone. Of course, is going to have an effect, right? Whole protein is gone. What kind of mutation is this? It's a base substitution that leads to a nonsense mutation. Okay? Any question? All right, let's go again. and you want to chop it up. You always want to chop it up. Now, when you chop it up into triplets, you notice something. Like before in the wild type, in the original, it's evenly divided by 3, right? But now when I chop it up here, I have like an extra one here. Because I have an extra one here, I mean, the T is not necessarily the extra one that was added, but then what I mean is like I have an extra nucleotide that caused me not to be able to evenly divide it into 3s, right? So this tells me something was inserted, and we have to find that extra nucleotide that got inserted. Where is the insertion? Find the extra nucleotide and tell me what it is. Does anybody see it?
Is it the G and like the fourth one from the front?
The first G, right? Yeah, that's right. There you go. That's the extra one. Right? You just compare it one by one. Right? TAC, TAC, ACC, GAC, right? Like so that's the G. And again, how do you know this is an insertion and not a base substitution? Because when you chop it up, I got a remainder, right? If you would, like I have an extra thing. So something was inserted. Okay? And then I copy it. I mean, not copy it, but then like I have to do the transcription. So okay. Now again, because we don't have too much time, I'm just gonna write it out for you. You look up AUG, you get MET, right? You look up CUG, you have LEU, GLU, Pro, and then LEU, LEU. So will there be likely an effect? Yes. Why? Well, look at this mutated sequence. There are two big problems here. Do you guys want to tell me what the problem is? Like if you compare the original sequence with this sequence, what's the problem?
There's a frame shift, so there's the extra G and the left out T.
That's right, but what about the protein? How's the protein being affected?
There's no TRP anymore.
Is it just TRP that's different?
And there's the pro now,
Are those
right?
the only two things that are different? I mean, you're on the right track, but
There's a lot of differences now.
that's what basically we made a completely different proteins, right? Didn't we? Look, other than the MET, everything else is different, right? And you were supposed to stop after the 1, 2, 3, 4, 5, after the fifth one, but because you frame the shift, sorry, you shift the frame because you shift the frame, it doesn't even stop. Like the stop codon doesn't even exist in the new frame. You just keep on going, and you make this long protein that is totally different than the original, right? So what's the problem? Insertion, which leads to frame shift. And the frame shift, because you change the reading frame, we have extensive. Extensive just means a lot of, right? Extensive missense mutation. Okay. And then like, just on a side note, no stop codon in the new frame. It just keeps going for some time. It keeps going until maybe there is some kind of stop codon later on. But eventually it's gonna stop, right? Eventually it's gonna stop because the mRNA transcript is not like, you know, it's not infinitely long, right? Like when you hit the poly A tail or something, then maybe it will stop. Is this okay? Now, maybe I'll quickly show you one more, and then you can do this. I'll of course post. I already post the answer actually online, so you can check later. But let's take a look at here. Okay, what's the first thing we do when you get a sequence? You chop it up. So 3, 3, 3, 3, 3, and now, you know, I was supposed to like chop it up again, but then like I'm missing something. So this tells you rather than, well, definitely is not a base substitution. Base substitution means I would have all the triplets in place. It's not insertion because we don't have like an extra one or something like that. We in fact missing one here, so it has to be a deletion. Now, some of you might ask, right? Like, well, maybe you have insertion of two things, but it's not true. Look at the original. We have 1, 2, 3, 4, 5, 6 codons, right? Here, I only have 1, 2, 3, 4, 5 codons, and then the last codon is missing one thing. So it has to be a deletion. Okay, deletion. And then you can figure out the rest. Now, I also want to tell you that when you are inserting or deleting a nucleotide, only the amino acids that comes afterwards are going to be affected. Okay, everything before the point of insertion or deletion, it's going to be fine. Now I want to show you another one here. This is totally not related. Okay, let's just say I have a coding strand here, just to keep it simple. Coding strand. Okay, totally making this up. Sorry, let me try it again. ATG TT ACCG hold on, that won't work. Okay, something like that, and then CCC GGT TAC TAG, sorry, no, C. I'm trying to do this in my head, that's why it's like so choppy. And then like that. Okay. So the mRNA is AUG, UUA. Everything is the same, right? Okay. Never mind what the amino acid sequence is. You can start, you can do that on your own. This is starting with ATG. Yeah, because it's coding, right? Coding is a DNA, right? That's why it's ATG. Okay. And then when you translate it to mRNA, it's AUG, right? Then it's a start codon. All right. So let's take a look at what happens here. What if I put a T here? I do an insertion of a T here. What's going to happen? Well, if I insert a T here, when I do the mRNA, that's like having a U here, right? So let's read this. If I'm the ribosome, let's read the code. So we'll read AUG, and then we'll bring the methionine. And then UUA, I think it's a lysine or something. It doesn't really matter. And then what's the next code? Instead of reading the GAG, because of the insertion, what's the next code that will be read by the ribosome? Now, because of frame shift, the next code that the ribosome will read is UGA. What is UGA? Stop, right? So you see, insertion can lead to frame shift. And frame shift can have different results. You can have extensive missense, okay, such as the example we did over here. Extensive missense just means a lot of mistakes, right? The whole thing is changed. But it's also possible to create a nonsense mutation if there is a new stop codon in the new frame. Okay. All right. Let's do a few more questions here. I think I have some in the PowerPoint. Let's do that. We have 3 examples here, 4 examples. Okay. Let's do them. This is back to the PowerPoint. Create a base substitution. Base substitution means you only change one nucleotide in the coding strand. This is the coding. That will result in a missense mutation. Missense mutation. So the easiest way to create a missense mutation, well, actually, you know what? Let's change it to mRNA first, because the codon chart is based on mRNA, right? It's easier if we look at the mRNA. So I give you the coding strand, you can just copy it. Right? All right. So how do I create a missense mutation? You can change anything you like. Like the easiest way to do it is just to change the first nucleotide. Okay. If you change the last one, it's okay too, but then like you have to make sure that they are not like repeats, right? But changing the first one or even the middle one is very likely to make it different. So you can change it to anything you want. You want to change it to like, G maybe? Okay, change it to G. So the original was CAG. So CAG, let's look at CAG. Right. CAG is GLN. That's the wild type. And then what about the mutation? Mutation is GAG now. So you look up GAG. That changes it to GLU. That's a missense mutation right there. You changed it. Okay. Now, do you have to do it here? No, you can do it. You can do it anywhere, really. I'll do another example here. I can change this to a C. So the original was GAU. You can look up GAU, which is aspartic acid. And the new one is GCU. GCU. It's alanine. That's a missense mutation.
I have a quick question.
Sure.
So the base substitution is just changing like one letter from what was already given, right?
That's right. One nucleotide, one letter. We're not inserting anything. We're not deleting anything. Okay, you try. This is the coding strand. And you probably want to write it as mRNA to make it easier. And I want a nonsense mutation. So we can copy this first. To create a nonsense mutation, I need to have one of these, right? UGA, UAA, or UAG, right? So it's you cannot do it anywhere. You have to find the right spot for it. Does any one of you have an answer for me? Where should I make the change so that I end up having one of these stop codons?
For the GAT near the end, you can turn it to oh wait, no, never mind.
No, okay, yeah. Roy, Roy, you said the third codon. Okay, what do we change it? How do we change the third codon? Change the first A to U. That's right. If you change this to U, then you have a UAG. That's a stop, isn't it? Right? So you will not make the rest of it. And what would the change look like in the coding DNA? Well, U corresponds to a T. That's one possibility, right? Is there another spot where this is possible? Does anybody see another answer?
The one on the end.
This one?
Yeah, it's already a stop one, but
Yeah, it's already stopped. So like, you know, we don't have to change it. Like even if we change it, it's not gonna be a nonsense. Right? Nonsense has to be second one.
Second
That's right.
codon.
And which letter do we change?
First letter.
That's right. We
C,
change it to
U.
That is right. So if I do this, UAG is also stop, right? So both are correct answers. And you don't have to do both of them, you just have to do one of them. Okay. Now we still have two more examples, but I want you to try these on your own. Yeah, I was gonna have the quiz on Tuesday. I think it might be too rush. Okay, I still want to do some more practice with you guys on Tuesday, like from the study guide. You work through it, right, yourself for the rest of the week first, and then we'll do more on Tuesday. I'll move the quiz to Thursday. Okay, the same day as the lab that is due. Tuesday, like I said, I'll do more examples with you. I will talk about the lab with you, and then you will have time to work on the lab. Okay. I can even push the lab to Friday, maybe. Okay. Let's do that. I'll change the quiz to Thursday next week, and then I will push the lab to Friday. All right. And then no class on Thursday. Okay? Thursday you can just spend, taking the quiz and then doing the lab. All right. Of course, I'll post all these changes on the eCentennial. Okay, but make sure you come on Tuesday because I'll explain how to do the lab. There are a few like calculations and math equations that I have to teach you, so that you will do better. Okay, that's it. Thank you. Enjoy the rest of your week. Bye.
Thank you, Professor. Bye.
Thank you, sir.